Good evening, and welcome to Argonne's virtual Out Loud lecture series. I am Leslie Crone, Argonne's Chief Communications Officer, and I'm pleased that so many of you have joined our forum tonight. Let me begin with some housekeeping remarks to help you get the most out of tonight's event. We recommend that you turn off any VPN for the best connection and experience. Your video and audio were automatically turned off when you joined the meeting, and the public chat has been disabled for this event. However, to ask questions at any time through the event, please use the Q&A module by clicking on the icon that looks like two talk bubbles. The moderators will answer as many questions live as time allows. You can also use the Q&A module to seek technical assistance. For example, if you can't hear the presenters or see the slide deck, please use the Q&A feature to let us know your issue, and a member of our team will connect with you to troubleshoot. And lastly, please note that this event is being recorded and will be posted for viewing at a later time. And by participating in this session, you consent to being recorded. Now, it is my pleasure to introduce Dr. Stephen Streifer, Deputy Laboratory Director for Science and Technology here at Argonne, and also the Interim Associate Laboratory Director for Photon Sciences, and the Director of the Advanced Photon Source. Stephen, please take it away.
Thank you, Leslie. Welcome to everyone to our virtual Out Loud lecture tonight. Tonight, we're going to focus on how COVID-19 behaves in our environment, specifically in the air we breathe and in our waterways and systems. As Leslie said, my name is Stephen Streifer, and I have the honor of serving as the Deputy Lab Director of Science and Technology here at Argonne National Laboratory. And as also Leslie mentioned, I'm Interim Associate Laboratory Director for Photon Sciences and Director of the Advanced Photon Source. We'd like to let you know that this year, Argonne is celebrating its 75th anniversary. Since 1946, we've been conducting cutting-edge basic and applied research for our country. Argonne started its history with nuclear energy. We trace our roots to the Manhattan Project with a founding mission to develop peaceful uses for nuclear power. Over the decades, we've expanded to what we are today, a multidisciplinary science and engineering research center accelerating our country's prosperity and security. We plan many events to celebrate 3/4 of a century of scientific excellence. Please look for announcements on our website and in your email inbox. We hope you can join us for upcoming celebrations throughout the year. Now, turning to our COVID-19 research, since the start of the pandemic, our scientific research has contributed to the fight against the novel coronavirus. The rapid development of the vaccines available now can be attributed in no small part to decades of research into infectious diseases that preceded the outbreak. This is a testament to the importance of strong support for basic scientific research. The National Lab Complex could not have deployed its science and technology workforce and facilities for this fight so swiftly without consistent investment over the years. A great example of this occurred actually at Argonne's Advanced Photon Source, or the APS as we call it, which was critical in providing early supporting research needed for vaccine development over about the last decade. The APS provides ultraviolet X-ray beams for research in almost all scientific disciplines. We use these beams to illuminate the structures of the virus and produce detailed pictures of the makeup of their proteins, which our partners then use to define effective drugs. Another example of how we sustain scientific investments preparing us to address COVID-19 is the use of supercomputers at the Argonne Leadership Computing Facility and other national laboratories to speed up the lengthy drug development process. These are only a couple of ways we're helping to mitigate the impact of the pandemic. We also advise state and local governments, built statistical models of the spread of the virus in local communities, and develop materials for more effective masks that could be cleaned and reused. Although much has been discovered, there remain many unknowns, and you'll hear a lot about that tonight, including understanding the behavior of COVID-19 in our environment. Since the onset of the pandemic, the public has sought guidance on how to live in a world with a virus that causes COVID-19. Argonne scientists have joined the global scientific community to answer these questions. In fact, our lab is a leader in this field. Scientists, as you'll hear tonight, in our Environmental Sciences Division are engaged in critical research examining how COVID-19 viral particles, so-called virions, behave in air and water. They're collaborating with 6 other national labs and many other organizations in both the public and private sector on this effort. Tonight, they'll share some additional findings with you, our virtual audience. To help lead these presentations, we have Christina Negri, Director of the Environmental Science Division. She'll serve as our moderator for tonight's Out Loud lecture. So thank you, Christina, for guiding the discussions, and please begin our program.
Thank you, Stephen, and good evening, everyone. Thank you for the introduction. I am the Director of Environmental Science Division here at Argonne, and before we get into our presentation, I will give you just a 5-minute overview of our environmental program at Argonne, because this is a foundation on which our work on the environmental aspects of SARS-CoV-2, the virus that causes COVID-19, is based. We have 7 different groups here that work in close connection with each other. The first one, Geospatial Computing and Sensing Group, brings advanced geospatial computational and sensing tools to environmental research. For example, we use computer vision to understand the collision of birds with solar facilities. In social cultural systems, archaeologists and anthropologists work to understand the threats from human activities and climate to our cultural heritage and offer ways to protect them. For climate research, we study planet Earth, and we have developed some of the highest resolution models of climate impacts at the regional and local scales. These models help find ways for communities to adapt and to become resilient as the climate changes and more extreme events are expected. The Radiological, Chemical, and Environmental Risk Group assesses the risks from radioactive or chemical substances to humans and the environment, and developed a code to use that is used worldwide to assess these risks. The Hydrology, Remediation, and Risk-Based Restoration Group characterizes, models, and finds ways to remediate contaminated water. For example, we work with the United States Department of Agriculture to clean up contaminated groundwater in Kansas, Nebraska, and Missouri. The Ecology team studies the impact of human activities on ecosystems and endangered species. For example, we look at how to improve ecosystem services and pollinator habitat at solar facilities. And we help find ways to manage electric hydropower in the West that minimizes impact to fish populations. Finally, last but not least, we operate the largest facility that's called Atmospheric Radiation Measurement, or ARM in short. This facility collects data on the atmosphere. Our scientists run radars, sondes, and other instruments to probe atmospheric conditions and make the data available to any researcher in the world that needs them. Given these capabilities, it's no surprise that we are interested in how a virus behaves in the environment. Before a virus enters a human body, it has to find its way to it, and that happens through our environment. So we share the environment with viruses. We breathe air, both indoors and outdoors. We use and discard water, and it's our interaction with the environment as well as viral behavior that drives our exposure, how much we are exposed to it. It is a solid knowledge of these interactions that allows us to protect ourselves. Plus, as you will hear later from Eugene Yan, if we understand this behavior, we can also gain early insights into the presence of viral threats in our communities. And these may make our job easier, as Eugene will say. So now I would like to introduce the 2 talented scientists who will speak about what we learned on COVID virus, SARS-CoV-2, in water and in air. We'll have tonight 2 speakers. Eugene Yan is a principal scientist and department head of the Hydrology, Remediation, Risk-Based Restoration department. He will speak about water behavior of the virus. And Rao Kotamarti, who is our senior scientist and department head for the Climate and Atmospheric Sciences department, will speak about the behavior in air. With that, Eugene, the floor is yours. Take it away. And when you're done, please pass on to Rao. Thank you.
Thank you, Christina. It is my pleasure to be here to share our recent study on virus transport in water. This research is contributed by many of our colleagues in Argonne and also at Northwestern University. Next slide, please. Now, first of all, we will ask a question about how viruses get into the water. So usually virus shedding after infections that could come from different locations, for example, from the hospital, quarantine center, seaport, airport, or residential houses. And those can go through sewer line to the wastewater. There is also some way goes through the stormwater. They pass through the farmer with the infected animals or farmland. They have untreated sludges. So those are some of likely way goes to wastewater. Now, it is important to understand once getting to the wastewater, what is the potential they move around? Where do they go? How do they behave? What is the potential pathway to our waterway and also to our groundwater? So this study, it is try to explore those questions. And mainly our approach for this study is try to use computational model to explore these behaviors. And before I go further, I want to emphasize that at the current time, we do not have enough data and evidence to indicate that COVID-19 could be transmitted through the water. But it is important to understand how virus could move in the water and also could be used for future pandemic and also bacteria. Next slide, please. So to understand the behavior in wastewater or in the surface water or groundwater, first of all, we need to know how much virus load into the water. So with increasing population infected, the virus load could increase to the environment and also particularly to the water. And those information, can we use it? Can we detect it first? And then can we use this information to supplement our prediction about future case trend for the virus, and also for our detection in particular area or for the early warnings. And once virus into the water, how virus transport in water, how fast they can go, and can they survive longer time and continue move to the next receptors. So those are questions we try to explore. Next slide, please. So first of all, many studies have looked at wastewater and collect the samples from wastewater. It is confirmed that virus did occur in wastewater. Through the RNA analysis, we found there is a concentration in the water and based on genetic information. So this is confirmation let us know that virus occur in the water. So next step is how do we use this information to look at prediction and the potential migration in the water. Next slide, please. So there is a many way once virus enters into water, they can go through sewer line, go to the wastewater treatment. But most of wastewater treatment method can stop virus further movement. But virus also could go through sewer overflow, combine the sewer overflow potentially to surface water, or through the leakage goes to potentially go to groundwater. So our question is, along these movements, what is the process will affect virus behavior? And what kind of condition is favorable to what virus move around with water? What the condition it's not? And on what kind of time scale virus can move? Because these are very important for us to understand those movements will be important, significant or not. Next slide, please. So there is a several study is going on, actually many studies going on for many communities. They test the wastewater and they found a concentration in wastewater. So here are two examples. One is in Boise, Idaho, the other in Los Angeles. So both show that virus in the wastewater. You can also see the Los Angeles case, the concentration of virus is 10 times higher than what we detect in Boise, Idaho. the shade, the blue band, this is the concentration of the virus. In a shaded area, it is a case confirmed by clinical testing. And then red line is hospitalization number for each time, for each day. So those information can see virus concentration, it's very similar behavior curve up and down with the case development and the hospitalization. And sometimes we also look at the concentration in wastewater, they are maybe same or few days ahead of actual case. And in addition, the actual case situation, the reporting time is usually 3 to 1 week later. So it is behind. So virus testing, if we can do the quick testing for the wastewater, we'll be able to predict the trend. So these additional, so this has become a powerful tool. Additionally, they also can include the symptomatic and asymptomatic case. So that has more broad interpretations. And this also can help us to identify hotspots and some early warnings. Next slide, please. So now, once the virus in the water, and especially if it moves through groundwater and it goes through contact with porous soil, what kind of process will it be? So virus, as we know, SARS-CoV-2, they have the shell. It's formed by different types of the proteins. So protein will show different electrical charge based on pH value in water. So for the low pH, protein will show the positive charge and then attach to the negative sides of soil surface. For the high pH, protein will show negative charge will attach to positive sides of the surface. So this mechanism helps us to understand how viruses attach on the surface, how they detach the surface. So we can formulate this process to predict their behavior. Next slide, please. So another important factor is how fast the virus can move in the groundwater. So we evaluate different types of the soil. So for the gravel, usually have a larger pore, they move fast. And we also found the fracture rocks, they even move faster because virus is very small particles. They can move through small fissures and particles, the fractures quickly. Next slide, please. Another important things we also need to look at is how virus will be inactivated. So as we know, high temperature, virus will survive a short time and a few hours. Here is the groundwater temperature in the nation. Groundwater, it's different from, the temperature is different from air temperature. It is cooler than air temperature. So the distribution, you can see the southern part has slightly high temperature and the northern part has a lower temperature. But overall, they are lower than air temperature. So their survival time is longer. Many studies also show that virus can also survive longer in clean water and shorter in wastewater. But that is good news because wastewater is a source. They have a shorter time to survive. Next slide, please. So we consider all these processes and then formulate them to develop a model, use the model in the numerical engine developed at Department of Energy. We call the P-Filtran model. So integrate all these processes, and then we use this model to evaluate what is the virus travel time, and what is the virus viability and the concentration, and also what is the effect of environment could impact virus movement and the travel. And for example, different soils, a different water chemistry, and a different setting, like what kind of leakage and a pumping well. So those will affect the virus final travel to any destination or potential receptors. Next slide, please. So we use this model, first of all, we need to validate our model. So there is several studies conducted in lab. So we use the lab data to do the benchmark testing to validate our 1D model to see our model can predict very well and fit to the lab data. And the next is the second one. We look at a 2D to evaluate what is the effect of a different parameter, as we discussed. If they flow, just a flow with the water, it's the top left figure, or they have inactivation attachment, that's a low right figure. You can see with attachment inactivation, they have a lower concentration and also move slow. So next, we move this to the three-dimensional model, and we based on some scenario to look at a leaking source and potential pumping wells. So we run solid simulations to look at what is the common situations, what condition we need to worry, what condition we don't need to worry. So based on our simulation, we found this is a gravel case, and they move from gravel, even if it's fast, it's still, they cannot reach far enough, usually less than 100 meters. But if they go to fractures, they can move fast. But another condition is we also look at, it is what is the depth of the groundwater. If groundwater depth deeper than 5 meters, which is 15 feet, and we don't need to worry about even for the fractures. Next slide, please. So in addition to groundwater model developments, we also develop a surface water. So this model is particular for Chicago waterway. So we based on Chicago waterway physical structure and build a model. And then we use 50 years of data, flow data and chemical data and water quality data to evaluate these waterway models. So here is one validation show here is we look at the fecal index bacteria. So our model can simulate very well to indicate how fecal index bacteria moves through this waterway. So we use this model will be next step to look at SARS-CoV-2, how they inactivate from sunlight and then build into the model to break it in case if it happens in the waterway, how it goes. Next slide, please. So as we discuss all the tools we developed, so we can combine together to help us to monitor our community, monitor our environment, particularly for the water. So first thing is we look at waste concentration in wastewater. We can predict what is the trend, what is the early warning, and which area could be get into the high infected populations, so which is a hotspot we can detect. So based on this, and then we can decide which area we need to move further. We need to look at more details, so we can run groundwater model to look at how move in the subsurface, in the soil, and we also can use surface water to look at how they move in the surface water, which area we need to pay attention, and that could guide us for the future sampling, and if necessary, also inform us how to do mitigations. So with that, so for this research, we develop an entire tool which is potentially very powerful for us quickly to monitor our future and pandemic and also application for any other virus or bacterial movement in water. So with that, I would like to turn over to Rob Kotomasi, and he will discuss about virus move in air.
Thank you, Eugene. I would like to start by introducing my team that worked on most of the project. So Ramesh Balakrishnan, Som Datta, Alex Obapko are the fluid dynamics that worked on the project. Myself and Yan Fang are atmospheric and aerosol scientists. So next slide, please. So this has been a lot of confusion about particles in the air. So I think it's good to start by cleaning up, or at least trying to understand some of the terminology that we have been exposed over the past year. Everything that comes from our body is essentially like breathing, talking, coughing, and sneezing. We refer to all these things as particles. They range from a submicron to thousands of microns. So what are microns? So you could ask the question, what do you mean by a micron? The panel in the middle actually shows a micron in some commonly understood things, like human hair is about 50 to 70 micron. And particle matter, PM 2.5, for example, you might have heard in relation to air pollution. So PM 2.5 particles are mostly produced by combustion, like a diesel engine burning, diesel burning, and the diesel engine would produce particles less than 2.5. Dust, pollen, and mold generally tend to be around 10 microns or less. So this is the kind of range of to understand what a micron is. So given that over the past year, it has become obvious that we need to divide the large space that particles occupy, like from less than a micron to all the way to 1,000 micron, into two boxes so that we can develop targeted strategies for controlling COVID. So we start by calling all particles greater than 10 microns. People use different ones. There is no standard for this. Some people call everything over 5 microns as droplets, but let us stick to 10 microns here. Everything greater than 10 micron droplet as shown on the right, and those that are less than 10 microns as shown on the left are called aerosols. The one greater than 10 microns are called droplets. So droplets are produced primarily by coughing and sneezing. And the size for this range from about tens of microns to thousands of microns. So if you want to compare the size of a sneeze and a cough to something that we understand, a raindrop may be around 2,000 microns in size. So that is falling from the sky. A sneeze can produce up to 1,000 micron drop. So we are pretty close to being a moderate shower when you get under a sneeze. So these are mostly small, so they are comparable size to that. So most of the particles are produced in the size range, but you also get some smaller particles when you sneeze and cough. The primary reason to put this in a separate box is that these particles, these droplets don't travel well. After they come out of our body, they quickly fall to the ground in less than 1 to 3 feet. So the mode of transportation for an aerosol is, for a droplet is different from an aerosol. Aerosols, on the other hand, are sizes of between less than a micron to 3 microns, can travel quite far. So they get suspended in the air and they move in the air for quite a distance. So if aerosols are the primary reason for transmitting the virus, then we need to take a different set of actions as compared to droplets. So next slide, please. So how is COVID spreading nowadays? So there is, if you look at the last 12, 13, 14 months, so there is enough evidence to suggest that COVID is primarily spreading through air. Most of the super spreader events that have been observed are in closed spaces, crowded spaces, or close contact settings, like people are talking and singing and things like that. So this has been COVID has not kind of uniformly spread. They spread in spurts or super spread events, and they tend to happen in these kinds of environments. And this gives us credence to the idea that it is spreading through air. So the next slide, please. So what I would like to do next is look at how these particle sizes and diameters change with various types of activities we perform. So let us look at breathing and talking as an example. The figure on the right shows particles, and on the left is the particle number density, number of particles that are emitted. So essentially, how many particles are emitted in a second. We have two panels, one for breathing and one for talking on both the figures. The bottom axis of the figure shows various ways we breathe and talk, and the vertical axis, for example, on the left side shows particle numbers, and on the right one shows the diameter of the particle. The data is shown as a box with whiskers going out at each end. So, for example, if you collect 100 people, sample 100 people breathing, the 100 samples are the 50 of the middle 50 of the sample is plotted as a white box there, and the median of the distribution is drawn by a red line. So keep the attention on your red line when I'm talking as I keep explaining this. Breathing through the nose produces the least number of particles per second, and fast breathing, like for example, if you're exercising or doing some intense activity, almost produces 3 times the number of particles as nose breathing. Mouth breathing produces almost twice the number of particles as nose breathing. the next block shows talking. So if you're talking slowly in a normal frequency produces maybe almost 3 times, 4 times the number of particles as breathing. And if you're talking loudly, then you almost produce 3 times more number of particles as you're talking quietly. So there's a big range in how you talk and how you breathe in the number of particles you produce. In the figure on the right, what you're seeing is the particle sizes produced by all types of breathing are about the same. They're about less than 1 micron when you're talking. They produce mostly small particles. And the particles become slightly bigger when you are talking. They are more than 1 micron. The important thing to remember from this slide is that all these activities produce small particles that fall under the aerosol size range, and they can stay in the air longer. So we are producing and talking and breathing, we are mostly producing the size of particles that can travel. So next slide, please. So how do we put all this information together to develop a better understanding? And the idea is that you want to do some mitigation or to take action so that we minimize our exposure to these particles, right? So at Argonne, we use world-class computing resources and computational scientists to develop computer model of aerosol movement. You can actually do this in a laboratory study. Our colleagues at Berkeley have instrumented a room with sensors and mannequins breathing aerosols to collect data. Our approach helps understand the data they're collecting and perform further studies. We take the various ways we expel stuff from our mouth and nose, as we have discussed before. And we also have a pretty good idea what is the size distribution, that is what kind of sizes we emit when we talk and cough and breathe. And the number of particles we produce, we call it emission. So we have a pretty good idea of emissions. Take these two and put in a space. And the space we selected for our numerical experiment is a classroom. There's a typical size classroom in Illinois, for example. So when we take that, we put that we put the classroom, it's designed in a computer, and the flow inside this classroom is solved using a computational fluid dynamics model called NEC 5000 to simulate the flow of air and particles in the classroom. So you may be asking, what is a computational fluid dynamics model? As you sit in your chair, you're surrounded by air and it is flowing in different directions depending on if you have heating on, the windows are open and things like that. So there are parcels of air moving around you at various speeds depending on various conditions and how fast you're moving your hands and things like that. The physical laws that describe how the air moves around your body and inside your room are codified by a set of mathematical equations known as Navier-Stokes equation. They are so complex that you need a computer to find a solution, and you need a pretty big computer in that. We include particles. So we have these computers and we have these models, and we do the simulation using these equations in a large computer. We include particles that can deposit and evaporate under these conditions. So the model also includes particles and evaporation. By solving this equation, we can find out where these particles move once out of the body, where do they deposit, and what effects environmental conditions like relative humidity and temperature have on the fate of these particles. Next slide, please. So we model a standard size classroom with a staggered desk spacing. So this is the classroom shown on the left here is an idealized classroom. The desks there are staggered. They are about 13 desks. So this is like the 6 feet apart that we are doing in Illinois right now, or many parts of the country. And the room size is about approximately the size of a classroom here. So to solve this, the mathematical equation on a computer for a room like this, you have to digitize the room configuration. We use 250 million points in the rooms at which we calculate the fluid flow at about every 100th of a second. So these 250 million points, we call it a mesh. It is similar to a pixel on your phone. The higher the pixel density, the better the resolution, right? The only difference with this is that these pixels are actually 3D. They have a volume in that instead of just a 2D pixel that you're used to on a cell phone, for example. Solving this equation on a big computer available at the Department of Energy, Argonne can take up to a few days to a week to just do about 15 minutes of real-time simulation. So it is pretty expensive to do and takes a lot of effort. On the right, you actually see an animation showing air moving from a HVAC system. So essentially what this is showing is that at the top corner of the figure at the back, there's a cassette type HVAC system where the air is coming in, and the air can go out through the front through a door. The red parts of the figure are shown as where we have a lot of turbulence. Essentially, turbulence means the air parcels, they are very agitated and they're mixing, they're hitting each other and mixing very hard. And this very turbulent part of the flow comes back from the wall and going out of the door. And then there is a lower velocity particles and then the even lower velocity particles, which are blue. And then there are parts of the room where you don't see these velocity arrows. What this means is that at these parts of the room, there is the air is stagnant. There is not much mixing going on. So one of the things we know from doing simulation like that is that if you have stagnant areas in a flow like this, particles tend to accumulate in those regions. So you could potentially have higher number of particles accumulating in the regions where there is no flow. So these are, this of course is only true for this HVAC system located as shown in the figure, and other ways, there are other ways of bringing air into the room and getting it to it can change the flow. A simple mitigation for this would be to open a window, for example, at the back of the room, or a heater or fans in the stagnant zones to mix things up. So when I'm talking, I'm just saying that mixing is better. So they're saying that mixing, dilution is the solution. Essentially, if you dilute something, let's say you have a glass of water and you put some salt in it, and if you don't mix and drink it, the water will be fine until you hit the salt, and then you get this big shot of salt, right? So if you mix it, it becomes more uniform, and the second thing is it reduces the concentration throughout the glass, for example. So our idea is to mix it so that no one person is getting hit by these particles or more exposed, and it is mixing and it's diluting, and the effects are less. So next slide, please. So in the previous slide, what we did is the air entering is at the same temperature as the room temperature. Here we actually put the air about 5 degrees centigrade hotter than the air in the room. This would be something like a winter condition in Chicago, where the air is coming, where you're heating the room, right? So what happens is under this condition, the warmer air actually stays near to the ceiling, and we call it stratification. Most of the turbulence is actually confined to the top of the room, and it goes through the door. So there is the lower parts of the room that don't get mixed so well, and you have to take mitigation actions to mix this air. One thing would be to have a floorboard heaters instead of putting in air from the top, or some way of other ways of removing air from the room that mixes it more. So next slide, please. The second experiment we did is we put air that is 5 degrees cooler than the air in the room. What happens is when you do that, the air in the room quickly descends to the bottom of the room and hits the floor. So when it happens, most of the aerosol particles in the room actually go very close to the floor, and because of all the air is descending to the floor. In a study that I mentioned at Berkeley, they have actually seen that a lot of the particles, when the temperature is colder, the incoming air, they collect near the floor of the room. So that's an interesting finding from this study. Next slide, please. So till now we talked about only particles that are from air moving. So let us take a quick look at how particles behave. This is a cough. Somebody is coughing at y equal to 0 on the y-axis. So that's a level the particles are coming. There's somebody standing there and coughing. So this comes out as a jet. And as you know, the right side of the figure shows the size of the particle. The left side shows the particle temperature. As we all know, our body temperature is higher than the ambient temperature most of the time. So the particles coming out from our body is hotter than the surrounding air. So it starts to cool down to the ambient air. The way you cool down to the ambient air is obviously evaporation. So when the particle evaporates, it becomes smaller. So we have a particle temperature and a size figures in here. So particles released here are range from about 1 micron to 50 microns. The bigger micron particles don't have enough time to equilibrate with or cool to the temperature of the room. They quickly settle to the ground before they even reach the temperature of the room. The smaller particles, the aerosols that are in the range of 0 to 5 or 5 to 10 microns, they are floating in this air and they have time to cool down. So once they cool down, they actually, because they are cooling down, they move further because their size is becoming smaller. So that is shown on the figure on the right, and the left it shows which particles are actually cooling down to the room temperature as time goes on. This is a an important thing to realize that the environmental effect is important. And the other thing here is that relative humidity in the room is kept at about 30%. So let's say the relative humidity in the room is much higher. Of course, you won't see this behavior because the particle cannot evaporate into a highly humidified room. If it is drier, you may see more particles evaporating. So this will have an effect on how far somebody is coughing or talking. The particles can go, is somewhat controlled by the environment in which you release these particles. Next slide, please. So with this study, I would kind of say that key findings are that when you do a simulation like that, we find that there are stagnant zones with very little flow. That is, as I said, these simulations were done with a refresh rate about 3 per hour. Essentially means that you're getting 3 new refills of air every 3 hours in this room. You can increase the refresh rate to 6, and it will look somewhat different because you're putting more air, and the air will be at higher speed because you're trying to fill the same room with in a faster number of refills, and you'll get that the turbulence in the room will increase, and the places where you see the stagnant zones will be smaller. Evaporation significantly affects the trajectory of the 4 to 32 micron particles, and it needs to represent it. So we need to consider evaporation when you're thinking about how these particles interact with the environment. So next slide, please. So the thing to remember is that we can do some common mitigation effects, and these kinds of studies help you develop those mitigation effects and understand why we are doing certain things. And also we have to realize that every classroom is different. The classroom, obviously, we don't have windows in this classroom. Let's say you have windows, the flow situation will be different. So the common mitigation strategies to avoid the kind of stagnant zones would be improve mixing with ventilation changes, essentially windows and things like that. Increase the room refresh rates from HVAC from 3 to 6, let's say, which is generally tends to be harder because you have to put a lot of money into that. But the other thing is that use of filters. So in this case, the air is not being pumped back into the HVAC system. Generally, about 20 to 30% of the air that is in the room goes back and recycled back into the room. If you do that, we find that if you put filters, actually it's very, very effective in removing the virus from the air. My colleagues at PNNL have done this study and have shown that it works very well. Use of fans and heaters can be used to clear stagnant zones. It's pretty useful to do that. In the long term, the idea is that if you use modeling like that, modeling studies like this, you can build better intake systems and output places where the air can go out, and design of the HVAC system to fit the rooms better and the classrooms more safer. So a lot of the mitigation efforts I've shown here are already implemented. One thing to realize is that classrooms in general have not been places where we have seen a lot of COVID transmission, but it is a classic. It is one of the cases when we started doing the study. Classroom, there was a big push to open the schools, and we were trying to figure out how understand the dynamics of fluid flow in the class, so that it would be helpful for people who are trying to open the schools. So I will stop there, and happy to take any questions. Thank you.
Thank you, Rao, and thank you, Eugene, for your presentations. A lot of work that had to happen in a short time, so very good. I have a few questions. We are going to start the Q&A question. People in the audience, if you have a questions, click the Q&A icon and type your question and submit it, and we'll do our best to answer them as long as we have time. So the first question is for Rao. Thinking of public schools, is it better to keep the windows open or closed as a classroom teacher? Also, in places of worship where people are singing without a mask, does the science show that it is a higher risk for attendees? Yeah,
I mean, in a classroom, obviously, I think keeping the windows open is absolutely a winner. That changes, essentially improving the ventilation in a classroom or any closed environment. So the thing to realize is that with COVID, there is hardly any case of COVID spreading in an open space when you're outside. So the more you make your indoor space look like an outdoor space, the less you would be experiencing the effects of this. So I think opening windows and increasing the ventilation is a good idea, great idea. And the second question was, I'm trying to remember the religious places, singing. Yeah. So the one case that I did not show the slide, there is concrete proof that it's one of the classic case studies, COVID early on, a super spreader event happened in a religious place where people were singing. And it's very early on, people were not wearing masks. And one infected person infected 87% of the people who were in that choir. So it does happen. Obviously, it is important to, when you are indoors, that's why the CDC still says indoor environment is one of the riskier places, and you should always try to wear a mask. As I shown earlier, the places where you get more post-COVID is indoor, and there are more concentration of people, and you're talking loud. So these are three whammies there. So you have to be careful in like a pub. That's one of the ideas, one of the things that was recommendation early on is that don't go to a pub or a bar, because everybody has to talk loud when they are in that situation, not without wearing a mask. And that was one of the advisories in the earlier, because as you've seen, speaking produces more particles than just breathing. So these are all important things to keep in mind.
Thank you. I am now moving to a question for Eugene. Could you describe the threats to humans or other organisms from coronavirus in water as opposed to respiratory droplets?
Yeah, for the coronavirus type, they are respiratory disease. So through the droplets and the transmit, it's much faster and short time durations. So through the water, especially if it goes through the virus shedding, through digestive system, and then goes to the water and the wastewater, and then this will take a longer time, even go through groundwater that could be survive longer, but it's a lot of factor will influence that. So through that word route, it's much difficult, and currently we don't have any evidence transmission goes through the water.
Thank you. A question for Rao. There was a recent study from MIT that said 6 feet rule is arbitrary. What can you say about that?
Well, that's why it's called it is a recommendation that we are following. So it is arbitrary if you look at the simulations we are doing in the sense that, but it is safer than not. We don't know whether it is 3 feet or 6 feet or 8 feet, right? So 6 feet seems like a reasonable number. Further away is better. Closer is not good. So that's, if you are within 2 to 3 feet, you actually get into the droplet scenario, which is worse, because the droplets can travel up to 1 to 2 feet from the next person to you. So 6 feet is considering that there are lighter particles, aerosol particles, that can travel that far. So it's a reasonable recommendation given the situation.
All right, Eugene, would wastewater treatment with UV light kill the virus?
Yes. So currently, all the treatment methods in the wastewater treatment plan and all their protocol is very effective, especially for SARS-CoV-2 and also several similar type of coronavirus. They are very susceptible for all these methods. So any like disinfections, UV light, high temperature will be very effective to kill all these virus.
Thank you, Rao.
Yep.
Compared to a classroom, can you speak a little bit about how safer or less safe are elevators, airplanes, and school buses?
Yeah, our next project is actually school bus modeling. So we are just starting doing that. So it is. So if you look at an elevator, Elevator is very interesting case. We really want to study an elevator because it's a confined space and opens at every floor, and it's essentially you're replenishing the entire air in that elevator every floor. Just boof, it opens and you're getting fresh air. So it's an interesting scenario to look at. There are filters in an elevator. My personal, we have not done enough studies. It would be less of a concern as compared to other situations. I would not. And if you are wearing a mask in an elevator and keeping your distance, I think it's not worse than any other place. As far as school buses, so one of my colleagues from Sandy has done a school bus. It depends a lot on whether the windows in the school bus are open. Like let's say you are traveling in a school bus with the windows open, it's pretty safe. There is absolutely your ventilation is sky high. But if you're opening, close all the doors, like in Chicago area in the winter, there are heaters, the heaters under the seats that we have, and they do circulate the air quite a bit. And the initial studies we have done suggest that actually it is fairly safe because the school bus is also stopping frequently. So the case we are studying is the bus stops and the door opens, and the air is from the outside, from the inside is going out, from the outside is coming in, and there is a HVAC system. So I think there are several ways the bus gets ventilated frequently. It's like a school bus or a city bus. I think it could be not as bad as we imagine it to be. That would be my initial guess based on, we have just started doing the modeling for that.
Thank you, Rao. Question for Eugene. How does the behavior of this virus in water compare to the behavior of influenza virus in water?
So for those virus, we do not have particular to look at the influenza virus, but the mechanism are similar. So in the transport in water, and we will look at how do they interact. For example, in the groundwater, how do they interact with the soil, and those attachment, detachment mechanism all affected by several things. One is the virus itself, the protein, how do they behave, and then under the water pH value, what kind of the water chemistry, and as also with soil green surface. So those are all interact to each other will affect your behavior. And the other thing is also for the inactivation, and there is also similar to the temperature, and if for the surface water and the sunlight, these are all will relatively affect that.
Thank you. Another question for you, Eugene. Is it safe to go to the beach this summer?
So I do not think currently we need to worry about anything on the beach. And so currently our study, it is to try to look at, because the several thing is when you have a pandemic, very high infect populations, that kind of situation, we will look at how much load in the water, and if there is any potential goes to any well. But based on current situation and the travel time and inactivation, especially in the hot seasons, the high temperature and the sunlight, those are factors really restrict the virus to move around and survive.
Great, Rao.
Yep.
For a classroom example, is there a way to purposely create an area of COVID concentration and pump the air out from that area?
Yeah. I think before I answer that, I want to say something, go back to a question before I forgot the airplanes case. The airplanes have, till now, they have a unique HVAC system in an airplane. Every few seats, there is a different way of air pumping in air and taking it out. Till now, there are no known cases of transmission within the aircraft that are not disputed. There are a few cases, but people are still struggling to find it's true or not. So going back to the whether I can concentrate all the aerosols, all the particles in one place and pump them out. That's an interesting idea. I haven't thought of that. You are, again, as I said, the flow situation is very, the one that we model has a specific constraints on it. So even if somebody walks in the room, the flow changes. So you cannot bet that this is good. The stagnant area is going to be constant, fixed for the entire like 40 minutes of your class. So that is the problem. I mean, if somebody is moving in or opening a door, these things change. So while it sounds like a great idea, you need some way to monitor the particles through the room to establish that actually that will work.
Thank you. Another question for you, Rao. Are the simulations done only via particle size, or are they also modeling the specific travel properties of COVID-19?
Yeah, the particles travel, do everything. They are moving with the air. They are falling down because of gravity. They have electrostatic effects. They have all kinds of effects. The entire physics of the particles is modeled. So these are Lagrangian particles in the sense that we release 1 million particles into the room of different sizes, going from 1 micron all the way to 50 to 80 micron, and we follow them through the room every 100th of a second, essentially. So yeah, it's a full calculation of everything, all the properties.
Wonderful. So another question is, can we conclude then that talking loud in a closed environment, such as a room mid-size or larger for several minutes, spreads the aerosol farther in the air as opposed to quiet talking, and this in turn increases the potential risk of exposure and infection?
Yes, without doubt. I mean, if you're doing that without wearing a mask. So somebody else also asked this question, what is the usefulness of a mask? So if you are wearing an N95 kind of mask, right? So that cuts everything from about 0.3 micron above, almost 95% of the COVID particle that is in an aerosol supposed to be around 1.1 micron. So it can stop most of the COVID particles if you're wearing an N95. So wearing a mask in an environment like that, when you're talking loud, is actually very good. An N95 mask is very good. If you're wearing one of those masks that are what the surgeon's mask or whatever it's called, the blue one, that only drops particles from 5 micron and above. It's actually designed for droplets, not so much for aerosols. So you have to be careful what you're doing, what you're wearing. But yeah, if you don't wear anything, there's some infected person and you're talking loud, these are the conditions for spreading, creating a super spreader event if there are enough people in the room.
So question for either of two, does the transmission rate vary with the ambient temperature of either air or water? Maybe you want to start with Eugene?
Yeah, for the transmission, there's several studies in the lab. We look at coronavirus type, which is similar to the SARS-2. So for these, we look at the data shows for the temperature above room temperature above, that survival rate could be within a few hours to a day to less a day. But for the groundwater temperature below to 10 degree or 10 to 5 degree, that survival rate could be increased to many days, even to the 100 up to the 100 days. So that low temperature could have. The other thing is related to the water. It is also, as I mentioned before, if the water is clean and the virus survives longer. If water is like a wastewater, there's a lot of chemical in it or a lot of other components, compounds in it, that virus tend to live shorter. And the other thing is also I want to, before I forgot that when I answer that question about beach water, what I'm looking at is the pathways through wastewater to beach water that we do not need to worry about. But on the other hand, at the same time, if we have crowded people all in the beach, that's a different story. So this also can be, so we still need to watch careful about rule and any transmissions through human to human at the beach. So that's another thing. So we need to watch. So we need to follow the CDC's guideline for that.
For air, I think the temperature and relative humidity are kind of connected. So if you have a high temperature and low relative humidity, the particles can travel faster. I'm not a virologist, so I don't understand much about what viruses do in a particle, but I can tell you how far they will go, right? If there is a virus on the particle and the temperature is high and you have a very dry air, this evaporation on the aerosol particles in particular, they can travel further. So temperature has an effect in that sense, that it increases the lifetime of that, or the travel time of the amount of time that can suspend in the air. Yeah.
All right. Eugene, could there be more potential risk of infection from shedded virus in bottled water? Because it has been reported that not all bottled water companies subject their bottled water to the same level of sanitization and disinfection as do public water utilities.
Okay, so first of all, I want to say that so currently, what our evaluation it is look at any water route through the wastewater, potential to the groundwater or to the surface water. So based on our current study, that it's only very few conditions that is the potential pathway for that. So if we look at the bottled water, that's mostly it's from surface water, and for most current situation, we haven't found any evidence in the surface water or in the groundwater. But our study is try to explore the behavior and to find what potential high concentration. So our study it is for a next step, because this could help us to guide, do we need to invest any potential vulnerable area to look at further. So currently no evidence to indicate any water is contaminated by virus and at the concentration that will affect people.
Rao, are HVAC design standards likely to be modified to reflect the findings of the research prompted by COVID?
That's one of the hottest areas of research for all those the guys working on MERVs. So the study I mentioned from PNNL used the MERV 11 for their in their HVAC system. It worked fairly well. Actually, it worked really well. It removed most of the particles in the air at the sizes that carry their thing. So I think it's there could be more improvements on them, but I think what we have right now probably will work.
Right, Rao, another question. I see many people wearing transparent face shields. Some deflect the breath upward, some downward. I suspect that neither has much effect in stopping particle spread, either to wearer or to a bystander. Is that correct? So
I think those were designed for droplets. So like you sneeze and cough, they can stop, like they can, the droplets, as I said, they're fairly big. They're as big as a raindrop if you're sneezing hard. So they can stick to that shield and drop down to the floor. So you're safe. But if it is talking and breathing and you're emitting the smaller particles, they can flow around that thing and keep going. So I don't think it is a very effective way to control them. I think you still have to wear a mask of some sort. If you want to wear that, it's a second level of protection, I think. That's because as I said, you produce particles when you're talking and speaking. And they tend to be really small.
All right, so Eugene, wastewater in dormitories has been used for early detection of COVID. What else can such systems detect?
What's that last? What else? What is the last question? Yeah.
Yeah. What else can such systems detect? When they looked at wastewater from dormitories that has been used for COVID detection, what else can these? What this approach could be detecting as well?
Yeah. So that's actually I see the several studies in the on the campus. They look at a dormitory, and it is reported. It is very effective way to look at. So this is also currently we are study continue with Northwestern University and also University of Illinois in Chicago area. We continue to look at that. So there's several questions we need to further answer is, so what detection level we can find? Because if you have only a small number of the infected people, you may not be able to detect. So if a detection limit, it's above that detection limit, so that could be a very powerful tool because we don't need to. A lot of people, young people, may not have the symptoms, and then they are actually infected. But so this tool can tell us not just symptomatic people and also asymptomatic people. So this is very powerful to do that. Yeah, that's I think that's.
Yeah, as you may have also heard, these methods have also been used to detect like substance abuse, like illicit drugs used in communities and things like that, right?
Yeah, so if we expand the area outside of virus, there's other things we can also use this for the detection. So the drug use, that's example that is already applied to some of the city in Arizona, and also some chemical use also can be detected through this system. So that's kind of identify spatial distribution, what either drug or chemicals, how do they distribute? That's kind of valuable for community.
All right, and Eugene, while you are talking, is there a difference between ocean saltwater and freshwater, such as Lake Michigan?
So the Michigan is freshwater. So as we know, so it's a large in the United States, the largest freshwater sources in the Michigan. So in also the most of the groundwater discharged to Michigan also it's fresh. Do not have any, as we know, have high salt. That's different from other areas. And there is, if we go to the southwest in the western area, there is a large reservoir for the salt water. I'm not sure that answers the question too.
Thank you. Rao, could you quickly repeat the relationship between quicker rates of evaporation, higher air temperature, and longer viability of the coronavirus?
Again, I don't know about the viability. The particles that of the size that can carry the coronavirus, the aerosols, will travel longer distances. So they have a potential to be in the air for longer. And if they have virus particles in them, then there's a probability of getting infected. I don't know whether the virus itself becomes more active once it gets drier. That's probably not a relation that I'm talking about. I'm thinking about how long does these aerosol particles last in the atmosphere. So that way, when you have a higher temperature evaporation, you see particles going further.
Thank you. And another question for you: in an office space, is it safe for people to take off masks if they are in small conference rooms with the door closed?
Well, say again.
Is it safe for people to take their masks off in small conference rooms with the door closed?
Well, if not everybody is, if you don't know they don't have an infection, if they have taken the vaccines, I think it's, as the CDC says, it's fine that we know we are with people whom we know. But if you don't know who the population in that room is, I don't think it's safe even at this time, because we don't know who could still be carrying the disease. So probably not a good idea.
Eugene, is there a risk of contaminated groundwater affecting our food, either through crops or through livestock?
So currently, I want to emphasize again, we do not find any virus contaminate the groundwater. And this study also help us to understand what kind of conditions and we don't need to worry about virus contamination. So we did thousands of simulation for most of the case and the most type of soil. We do not need to worry because the travel time is too long for the virus. So it's difficult to survive for them. And only very, very real situation is like a highly fractured situation. And also an additional barrier is if groundwater depth is high, it is difficult for virus go down to that deep depth. So for most situations, we don't see any favorable condition for virus survive in groundwater.
Thank you. A question, I believe this is a question from a kid. Do you think that it is wise to use a large high-rise building pool when I am not vaccinated as a kid? I mean, that's probably outside of our domain to know. So
yeah,
it's really hard. And
while they're thinking, Christina, maybe one more question, and then we're going to wrap it up.
all right. So how about public restrooms? How contaminated are they?
so the classic way we thought this virus spreads is essentially by contact. So if you think like that, then the public restrooms would be very dangerous. But for this particular virus, it seems to spread by air more than by contact. So if you're wearing masks and stuff like that, if you're safe with an N95 mask, and you should be okay. I wouldn't be freaking out.
Wonderful. Eugene, Rao, and everybody who provided question and listened in, thank you very, very, very much for your participation. I would also like to take the time to thank the National Virtual Biotechnology Laboratory for funding this research. And just as this is all the time we have for tonight, I will turn back the program to Leslie.
Well, thank you, Christina. And again, thank you to Stephen Streifer, to Raul, to Eugene for your remarks, your presentations, and diligently answering all those questions. It's wonderful engagement from our audience. We love to hear your questions and hope we've been able to answer them as best we could in the time that we have. So as I mentioned at the start, this event was recorded, is being recorded, and will be emailed to you tomorrow so that you can listen again or share it with your colleagues, your friends, and your family. Our next Out Loud lecture will be held on June 30th. It's a very special day. It is the eve of Argonne's 75th anniversary. And we will mark this milestone with a special Out Loud, during which we will look at the next 75 years of discovery and innovation through the eyes of our Argonne scientists. So please save the date and look for an email invitation with additional details to come. And with that, we hope you enjoyed your virtual visit with Argonne this evening. Have a good night.